We don't study any human cells, we study worm sperm from roundworms, which strangely enough crawl around just like various sorts of cells in our body crawl around. Some of our cells are pretty complicated, so basically if you want to figure out how a cell crawls, it's good to start out with a simple one, like say for example if you wanted to understand how an automobile works, it'd be easier to study a Volkswagen than say a Lamborghini. So we're basically studying the Volkswagen of the cell crawling world. So there are two sorts of forces that are involved, and we're all pretty much in agreement how the pushing at the front works, but the stuff that goes on in the rear has been a little harder to get at. And so that's what we've been focusing on. There are the worms right there. And the current study is focused on how the rear part of the cell is pulled forward. The current thinking is that there's a protein called myosin. Now myosin is more familiar as the protein that powers muscle contraction. And so a lot of people in this field think that myosin is the protein that does basically the same thing. It pulls the rear of the cell forward. But what we've found in the last 10 years or so, there have been lots and lots of reports coming out of cells that are able to crawl perfectly well when their myosin function is compromised or inhibited. My postdoc, Katsuya Shimabukuro, devised a way that he could reconstitute this motility in the test tube or under the microscope. And by watching this reconstituted system and taking it apart in various ways and looking at it with an electron microscope, he was able to figure out a plausible mechanism using this worm sperm experimental system, a plausible mechanism for how to pull the rear of the cell forward without using a motor protein like myosin. This whole body of research really would help us understand the basic principles underlying how we fight off infection, how we heal wounds, how we remodel our bones, how our neurons connect up with their targets, and how cancers metastasize.